Most health advice centers on what you eat, the protein ratios, the micronutrient profiles, the quality of fat and carbohydrate sources. But there is a biological repair process that operates largely outside the domain of nutritional composition, and it may be among the most consequential tool for long-term cellular health available to you. That process is called autophagy. It is not primarily activated by consuming specific nutrients. It is substantially upregulated by the deliberate absence of food. Most people believe their longevity is determined by what they eat. The evidence suggests it is equally shaped by how often they stop. Your body, during extended periods without caloric intake, does not enter a passive waiting state. It initiates a precise genetically encoded program of cellular maintenance that research is now connecting to longevity, disease prevention, and metabolic function in ways that dietary composition alone cannot fully replicate. Human biology did not develop under conditions of continuous food availability. For the vast majority of human evolutionary history, extended periods of food scarcity were a routine feature of existence. The cellular machinery inside your body was shaped by that environment over hundreds of thousands of years. When food is consistently absent, depending on continuous external fuel to sustain essential functions would have been a fatal vulnerability. So human cells evolved the capacity to dismantle their own damaged and surplus internal structures, recover the molecular raw materials from those structures, and repurpose those materials to sustain critical cellular operations. This is not a crisis response or a state of damage. It is a deeply conserved biological program present in organisms from single-cell yeast to humans, and it is strongly promoted by the absence of caloric input. Though, it is also influenced by other factors including cellular energy, status, physical exertion, and the overall metabolic environment. To understand how autophagy is regulated, you need to understand two central signaling proteins. The first is called mTOR, which stands for Mechanistic Target of Rapamycin. mTOR functions as the cell's primary growth activating complex. When amino acids and glucose are present, mTOR is active. Active mTOR promotes protein synthesis, cellular growth, and cell division. And critically, active mTOR suppresses autophagy. It is the molecular state that locks a cell into an anabolic or building mode. The second protein is called AMPK, which stands for adenosine monophosphate activated protein kinase. AMPK is the cell's energy sensing enzyme. When cellular energy levels drop, as they do during fasting, AMPK becomes active. Activated AMPK produces two immediately relevant effects. It inhibits mTOR, removing the suppression that mTOR was applying to the autophagy pathway, and it directly activates a protein called ULK1, which is a signaling enzyme that initiates the physical construction of the structures through which autophagy operates. Those structures are called autophagosomes. An autophagosome is a double membrane vesicle that forms inside the cell and engulfs damaged proteins, dysfunctional organelles, and surplus cellular material. The autophagosome then fuses with a lysosome, which is a cellular organelle that contains digestive enzymes. Those enzymes break down the engulfed contents into their molecular components, including amino acids recovered from degraded proteins, fatty acids recovered from lipid membranes, and nucleotides recovered from damaged genetic material. Those components are released back into the cellular interior to be used for energy production or for constructing new functional structures. This is the recycling mechanism described in mechanical terms. Damaged and surplus cellular infrastructure is systematically dismantled, and the recovered molecular materials are reintegrated into the cell's active supply. Let that settle. Your cell is not idling while you fast. It is selectively breaking down damaged components and rebuilding from what it recovers. The progression of this process during a fast follows a general pattern that research has begun to characterize, though precise timing varies between individuals, tissues, and metabolic states. In the typical fed state, blood glucose and insulin are elevated, mTOR remains active, and autophagy is suppressed. As hours pass after the last caloric intake, blood glucose declines, insulin falls, and mTOR activity begins to reduce. After approximately 12 to 16 hours of fasting, the biological signals that favor autophagy induction typically begin to emerge. AMPK activity rises, mTOR inhibition takes hold, and autophagic activity increases across multiple cell types. It is worth noting that measuring autophagic flux directly in humans is technically difficult. Much of what we know about timing in people is inferred from indirect markers, and the threshold will differ depending on the individual's baseline metabolic state. As fasting extends toward and beyond 24 hours, research on healthy adults suggests the autophagic environment continues to deepen as glycogen depletion becomes more complete and systemic. IGF-1 suppression increases. Research on extended fasting beyond 24 hours suggests further autophagic induction at this duration, but frequency, timing, and individual suitability vary considerably and are not appropriate as a general recommendation without medical assessment. For protocols extending toward 48 hours, the autophagic environment deepens further still as the metabolic shift toward fatty acid oxidation and ketone production becomes more comprehensive. But protocols at this duration carry increasing physiological demands and fall outside the scope of general public recommendation without direct medical supervision. The core principle that this timeline illustrates is worth stating directly. Your cells contain a genetically encoded maintenance and repair program, and caloric deprivation is one of the most powerful known inputs for activating it. Dietary composition matters, but the periods in which your body operates without food are a direct variable in whether the conditions for that program are being met. The scientific legitimacy of autophagy as a fundamental biological mechanism is established at the highest level of institutional recognition. In 2016, the Nobel Committee awarded the Nobel Prize in Physiology or Medicine to Yoshinori Ohsumi of the Tokyo Institute of Technology for his discoveries of the mechanisms underlying autophagy. Ohsumi's foundational work, conducted primarily in yeast and published in seminal cell biology papers in the early 1990s and later in major journals including Nature, identified the core set of autophagy-related genes, known as ATG genes, and characterized how those genes coordinate the formation and function of autophagosomes. These ATG genes are conserved across eukaryotic organisms, including humans, meaning the same core molecular machinery that Ohsumi identified in yeast operates inside your cells. The connection between autophagy and neurodegenerative disease is a well-documented area of ongoing research. Alzheimer's disease and Parkinson's disease are both characterized in part by the accumulation of misfolded or aggregated proteins inside neurons. In Alzheimer's disease, the progressive aggregation of tau protein and amyloid beta fragments within and around neurons contributes to neuronal dysfunction and cell death. In Parkinson's disease, toxic deposits of a protein called alpha-synuclein form inside the neurons of the substantia nigra, the brain region responsible for dopamine production. Autophagy, functioning normally, contributes to the degradation of these protein aggregates before they accumulate to damaging concentrations. Research reviewed in journals including Nature, Reviews Neuroscience has documented that reduced autophagic activity is associated with accelerated accumulation of these pathological proteins, and that supporting autophagy function represents a potential mechanism for maintaining neuronal proteostasis. the continuous cellular process of clearing damaged proteins to preserve the functional integrity of neurons over time. Put simply, autophagy is part of how your brain clears its own debris. When that process slows down, the debris accumulates. On the metabolic side, autophagy performs a specific and critical role in mitochondrial quality control. The selective autophagic degradation of damaged mitochondria is called mitophagy. Mitochondria produce the majority of cellular energy through oxidative phosphorylation. As mitochondria age or sustain damage, their energy production efficiency declines and they generate excess reactive oxygen species, which are chemically reactive molecules that cause oxidative damage to surrounding proteins, lipids, and DNA. Mitophagy removes these compromised mitochondria before their accumulation impairs the cell's overall energy metabolism. Research published in journals, including Cell and Molecular Cell, has established that impaired mitophagy is associated with reduced metabolic efficiency and elevated intracellular oxidative damage. Now, you might be wondering what all of this looks like in practical terms. The practical implication of this research is that the timing structure of your eating carries biological consequences that operate independently of what you eat. The periods in which your body operates without food are a direct input into the autophagic program. Time-restricted feeding, confining daily caloric intake to a window of 8 to 10 hours and maintaining a consistent fasting period of 14 to 16 hours each day has been studied in human populations and may create a metabolic environment favorable to autophagic activity, though direct evidence in humans is still developing. In practical terms, a 16-hour daily fast often means completing food intake by 8 p.m. and not eating again until noon the following day. For individuals who have established a consistent time-restricted eating pattern and are doing so under appropriate medical awareness, extended fasting periods may create conditions for more substantial autophagic activity than daily time restriction alone. However, this should not be treated as a universal recommendation. Extended fasting is not appropriate for everyone, and individual responses vary considerably depending on age, baseline health, medications, and hormonal status. These extended fasting periods require adequate hydration throughout. For individuals managing active metabolic conditions or taking regular medications, medical consultation before beginning this practice is essential. Women in particular should approach extended fasting protocols with additional caution, as prolonged caloric restriction can affect hormonal axes, including thyroid function and menstrual regularity, more significantly than in men. Anyone who is pregnant, breastfeeding, or has a history of disordered eating should not undertake extended fasting without direct medical supervision. Individuals under 18 should not undertake extended or restrictive fasting without guidance from a physician or registered dietitian, as the body is still developing. Exercise is an additional and independently significant variable, but the biological response depends heavily on the type of training. Sustained aerobic exercise, particularly steady-state endurance training, strongly activates AMPK and maintains mTOR suppression, driving a robust autophagic stimulus. Resistance training, by contrast, operates differently. While lifting weights creates an immediate metabolic demand, its primary biological directive during the recovery phase is the powerful activation of mTOR to drive muscle protein synthesis. Therefore, fasted endurance exercise may be one of the stronger practical signals for promoting an autophagy favorable metabolic state, though the response varies by individual, training status, duration, intensity, and safety considerations. It stacks the energy depletion of fasting with the metabolic demand of cardio, compounding both AMPK signals simultaneously. Some research suggests that aerobic exercise performed in a low energy or fasted state can increase markers associated with autophagy, but the human evidence remains limited and depends on the markers measured. The finding that threads through all of this research is the same. Your cells contain a maintenance and recycling program encoded into their biology by hundreds of millions of years of evolutionary pressure, and caloric deprivation is among the most powerful known signals for activating it. Dietary quality matters, but whether your cells are actively recycling damaged components or continuously suppressing the process because mTOR remains active depends substantially on how long you allow your body to operate without food. Yoshinori Osumi's Nobel Prize winning discoveries established that autophagy is not a peripheral cellular event. It is a core process that cells depend on for their own structural integrity. Enabling it is not a matter of adding something to your routine. It is a matter of removing the caloric signal that suppresses it for long enough to let the program run. If this mechanistic breakdown was useful, the next subject in this series covers cellular senescence, the process by which damaged cells stop dividing, remain metabolically active, and begin releasing inflammatory signals that accelerate aging in surrounding tissue. That video is already on this channel. Subscribe to the Longevity Matrix so you do not miss the continuation of this series.